Good morning, students. So today I will teach you about the topic controlled breeding experiments. Right? First of all, the meaning of controlled, it should be clear. The word itself, expressing, means the which is under the control, means which is we can do voluntarily. Means the you can understand this by the correlating again here the Mendel's topic, as the Mendel did the experiments with bee plants, and he did all his experiments voluntarily by keeping optimum conditions, and he got the desired results, right? So the meaning of controlled means the when the conditions are kept standardized, optimum conditions, and means the desired results are obtained, right? These experiments, breeding experiments, controlled breeding experiments, are done voluntarily, right? So these experiments can be done by two methods. First one is the AI, means the by the artificial insemination method, and second one is the MOET, means the multiple ovulation embryo transfer. So these two methods are usually done in controlled conditions. First I will take this one, that is the artificial insemination. In artificial insemination method, what is done? First of all, semen is taken from the superior male, right? And then it is injected into the reproductive tract of the female, right? So this semen, it can be taken and directly administered into the tract of the female, or it can be taken, then it can be frozen, and then it can be transported to the place where that particular female is housed, right? Means it can be preserved in the frozen state also, right? Then what is this advantage of this artificial insemination? Advantage is that fewer sperms for copulation, for the conception, right? Means for the successful conception, less number of sperms, they can solve the purpose. In natural conditions, what happens actually? Maximum sperms are released, but some of them, they are capable of fertilization. So here already viable sperms are kept, they are taken and then injected into the female, right? So fewer sperms, they will be needed here. Second one is there will be the less chances of the STDs, means the sexually transmitted diseases, right? For the few STDs, but not for the AIDS and herpes and all this hepatitis, right? Not herpes, sorry, hepatitis. So because these sexually transmitted diseases, they are also just spread by the sexual transmission, but when the semen is taken, then this incidence can be minimized, right? Then second one, the breeding, this controlled breeding experiment, that is the technology is the MOET, right? Multiple ovulation embryo transfer. Why this one? Because what happens actually, success rate of crossing mature male and female animal is very low, even though by this method, artificial insemination method. So then this technology is nowadays developed, right? So I will discuss in brief all this technique here in the form of this flow chart, and it will be easier for you to learn the concept, to clear the concept even, and it will be scoring also, right? So what happens here actually? First of all, one female is there. For example, I have taken here the Actually this MOET, it has been demonstrated in cows, sheep and rabbits and all buffaloes all. So I have taken here the example of cattle, right? So first of all, follicle stimulating hormone. This hormone is injected into the female. Okay, first of all you should clear the role of this hormone. Actually you have not still done the chapter unit that reproduction. So but in 11th class, I think in the last chapter, its functions of the hormones are given. So follicle stimulating hormone, it means the hormone responsible for the stimulating the follicles of the ovary to release eggs, right? That is the function of the follicle stimulating hormone. And in general what happens, one or two follicles, they mature at a time and then they release the eggs. As if I talk about the human beings, then one or two follicles, they usually mature once in this month. Then out of this one, this egg, it usually die, and one reach the this fallopian tube, right, where fertilization occurs. So this hormone is given to the female cattle here, that is why, for the maturation of the follicles. So here follicles will mature obviously, but difference will be what? Without hormone and by giving the hormone. By giving hormones, there will be the super ovulation. What does it mean? Super ovulation means the in maximum number eggs will be produced. How? Because maximum follicles, despite one or two follicles, there will be the maturation of the 6 to 8 follicles. So as a result, there will be the release of the 6 to 8 eggs, right, once. So that is the purpose of giving this follicle stimulating hormone, injecting this hormone into the that female. So after this what happened? After this, that female, that will be cross-mated with the any naturally by any superior quality male bull, or that female can be artificially inseminated. Two ways are there. One is artificial insemination and one is the naturally. Then fertilization will be there and zygote formation will be there. Not zygote, but zygotes formation will be there. Then what will happen after this? The cell division will be there. By the mitosis, there will be the formation of the embryo. Few celled stage is called the embryo. So several stages are there, but when it will reach the 8 to 32 celled stage, which is called the morula in case of human beings, right? Several stages are there, like the gastrula, blastula, morula, right? So this stage is called, when the embryo acquire 8 to 32 celled stage, that stage is called the morula stage, right? Then after this what happened? That embryos, they are removed non-surgically. Non-surgically means without doing the surgery. Clear? Then that embryos, then next step here, in this method two females are required. One that was given FSH first in the uterus of that female, where embryo just they get formed. That mother is called the genetic mother, because that mother is actually responsible for producing the eggs. And when these embryos, they are removed first of all non-surgically and then they transferred to the second female, that female is called the surrogate mother. You might have heard the term surrogacy. Surrogate mother will only receive the embryos, and she will she will provide all the essential conditions in her womb till birth. All that further development of that embryos till the birth, that will be in the womb of the surrogate mother. Clear? So this is the Usually this is the technique of MOET in the simpler steps. Clear? Now, I have told you earlier that it has been demonstrated in cows, sheep, rabbits, buffaloes, mare, etc. All these. Now, what are the advantages of this technique? Main first and one important advantage is that I told you only 1 or 2 eggs are produced usually, but here you can see by this the hormone, this such 6 to 8 eggs can be produced. So obviously increase herd size in short time, right? As more than one zygote can be implanted in one female. This is the advantage of this MOET technology, multiple ovulation embryo transfer technology, right? So this is all about this topic, controlled breeding experiments. Though very small topic, but very important topic. Okay, so clear this by reading again 2-3 times, and then this it will be definitely will be helpful for you. Okay, and thank you.